When one is exploring action potential, it is important to understand the basic structure of neurotransmitters and the synapse. To begin, the dendrites receive messages from other cells. Dendrites stem out from the cell body. Then the cell body sends messages down the axon, which is the action potential. On the presynaptic terminal, one will encounter calcium channels, and it opens up. Calcium comes in and causes the closed vesicles with acetylcholine. They migrate to the end of the terminal, open up, and transmit the acetylcholine. Once the messages reach the axon terminals, which is the presynaptic terminals, they combine with the postsynaptic terminals of another neuron.
A large number of voltage-gated ion channels are found in the cell membranes of neurons, which allow for the movement of sodium, potassium, and calcium into and out from the cell during an action potential. When a stimulus depolarizes the membrane to the minimum threshold of activation, sodium are the first ion channels to open. As sodium channels open, some sodium ions rapidly enter into the cell. Voltage-gated potassium channels open in response to the depolarization caused by sodium influx. The movement of potassium ions out from the cell repolarizes the membrane back to its negative resting potential. In the neuron, voltage-gated calcium channels are only found in presynaptic terminals. Activation of voltage-gated calcium channels allows the movement of calcium into the cell. Calcium triggers the release of stored acetylcholine from vesicles in the presynaptic cell. A synapse consists of the presynaptic terminal of one cell as opposed to the postsynaptic membrane of the next cell. The small gap between these terminals is called the synaptic cleft. Acetylcholine is released from the presynaptic terminal, then binds to and activates acetylcholine-gated sodium channels, which allows for sodium entry into the postsynaptic membrane. Acetylcholine is rapidly degraded and inactivated by acetylcholine esterase in the synapse.